Welcome back to our conversation on DNA replication. So far in this video series, we have talked about just some key features of DNA. Remember, it is a double-stranded molecule. Each strand is made up of nucleotides, and the two strands of nucleotides that make up the DNA molecule are anti-parallel and complementary to one another. We then looked at the big picture overview of DNA replication. And that's where one molecule of DNA, the two strands are going to be separated from one another and used as a template to create two new strands of DNA nucleotides. So then at the end of the process, there are two identical copies of DNA. So now we're going to start to zoom in on some of the details and introduce to you some more terminology, okay? So DNA replication begins at the origin of replication. And so these are just DNA sequences that are going to be kind of the starting point for separating those two strands, okay? Now, I'm going to show you the more complex process that happens in eukaryotes, but something similar, exactly similar, is happening in bacteria as far as we're concerned for this class. So prokaryotes, like bacteria, because they are smaller and more simple than eukaryotic cells, they only have one chromosome and they tend to have less DNA than eukaryotes. So they only need one single origin of replication. Because eukaryotes tend to be more complex, they have more DNA. So because this does need to happen in a quick, timely fashion, there are going to be multiple origins of replication along the piece of DNA. That way, this process can be done quickly. So what I'm showing you here is the more complex version of eukaryotes, where we have a big strand of DNA. And you'll notice that there are multiple dashes, and each dash represents an origin of replication. Trying to get there we go. So I abbreviate origin of replication on here as OOR, origin of replication. So again, I'm just showing you here with eukaryotes. And so at each of these origins of replication, the DNA is going to start to separate. And it does that because those hydrogen bonds are going to be broken. And so here's what happens. Do you see how they're starting to separate? So with this, we call these separated regions a replication bubble, right? Because it looks like a bubble in the DNA. Now, at the origin of replication, which is in the center of this bubble, DNA is going to occur bidirectionally, as you can see by these arrows. That means there are some proteins in the cell who are going to start creating that new strand of nucleotides going off to the left. And then there are proteins that are going to start making that new strand over here to the right. This is happening on both strands of the DNA molecule. So the blue arrow here is going to be that new strand that's built by the cell that I showed you in the previous video. So once this entire bubble has been used, it's been used to make that new strand of DNA. Well, now the cell needs to continue copying this other region, right? See where my mouse is? This part is still closed. It's not available to be copied. And so once the bubbles have been copied, the bubbles are going to expand a little bit, and the replication process, the copying process, is going to continue. And that's just going to keep happening until both strands have been used as a template and we have created two new strands of nucleotides, and we have two molecules of DNA. We say this process is semi-conservative. In the biology world, conservative means it's the same. So a lot of genes, like in humans, for instance, a lot of genes that we have now, generations of humans have had because they are critical for our survival. So those sequences stay the same. They are conserved over time. So when we apply that definition to conserved or conservative, this process is considered semi-conservative, because if you look at these two molecules, and I'm sorry, the captions are covering that bottom strand. I meant to move the image. But if you look at the two DNA molecules, each one has an orange strand of nucleotides, which represents the original starting strands from that parent DNA molecule. And then each of these new DNA molecules also has a newly created strand, which is shown by blue. So we say this process is semi-conservative because each DNA molecule has one original parent strand. So it's kind of stayed the same, right, as the starting molecule, because they each have one of those original strands, but it's not entirely the same because one of those strands are also newly made. Okay, so semi-conservative just means that each DNA molecule has one original strand of nucleotides and one newly created strand of nucleotides. Okay, so again, just to kind of add some labels, I'm a very visual person. We would say that here is one daughter molecule of DNA. Again, notice that first daughter molecule of DNA has one original parent strand of nucleotides and one newly created strand of nucleotides, so we say it's semi-conservative. Same thing for this second DNA molecule. Okay, in this case, the 3' to 5' is the original parent strand, and the 5' to 3' is the newly made strand. And again, just add some labels here, trying to be redundant on purpose to kind of drill these terms into your brain. So again, each DNA molecule has a parent strand and each has a newly made strand. Okay? So what we're going to do next is we're going to kind of zoom in on those replication bubbles that I just mentioned and introduce to you another term, which is replication fork. So let's check that out. So remember, we said that DNA replication starts at the origin of replication. And at those origins of replication, the DNA separates and we get a replication bubble. Okay, so that's what I've labeled first. All of these that you see here are replication bubbles. But for simplicity, as we continue to work through this process, and I tried to create these animations, I am not artistically skilled to keep drawing everything in this circular fashion. And so we're going to focus on just half of a bubble, which we call a replication fork, right? Because it looks kind of like a fork in the road, right? You're going this way, and then you can either go to the top or to the bottom, okay? So half of a replication bubble is known as a replication fork. Okay, so each of these bubbles, right, can be divided in half, and we can look at it as a fork. So just kind of more for simplicity of how you're going to see things going forward. Here's our replication bubble. Again, I've drawn the line down the middle, okay, so that each half is considered a replication fork. But again, my artistic skills are limited, and so instead of drawing everything in a curved fashion, I will be drawing it more linear. And so just here it is as more linear. And then showing you exactly what I'm going to be drawing, which is normally just this left hand half of the bubble. So I'm going to show you this left hand replication fork. Okay. All right, so let's end this video with an apply what you know. And these are things I am going to be able to expect you to do on knowledge checks, quizzes, tests, any form of assessment you're going to see in this class. So as always, I will quit telling you this in future videos. I recommend you pause it, do your best with just your brain. It's really important to try to make your brain retrieve information on its own, okay? So give it a shot. Don't leave this blank, right? You're doing this on your own. If you're confused or you're stuck, nobody's gonna know, okay? But give it a shot because you learn more from your mistakes than from not doing anything at all, okay? So I'm showing you a piece of DNA. You can see double-stranded. And I want you to sketch the two daughter molecules that will be made after DNA replication. And I do suggest you use two different colors, kind of like I've been doing, to show the original strand and the newly made strand. Okay, so I'm going to go ahead now and work through this. All right, so, oh, I really need to get used to this mouse. Sorry, y'all. All right, so I'm going to take this kind of one thing at a time. Where did it go? Where'd my other strand go? Wait, chihuahuas. Sorry, y'all. The other the strand should have been here. I don't know what happened to it. So we should have our starting DNA molecule. Notice the two strands should have separated, okay? And then we're going to use those as templates. So in my case, this green is going to be the newly made strand. So notice my newly made strand is opposite or anti-parallel to the starting blue strand. Okay, and the bases are complementary. G pairs with C, C pairs with G, A pairs with T, T pairs with A, so on and so forth. And then the same thing is going to be done to this bottom strand. So notice the newly made strand is anti-parallel, and again, our bases are complementary. But notice, y'all, the two DNA molecules are the same in terms of the directionality and sequence of the top and bottom strand for each one. Okay, so again, the parent strands in this case are the starting strands, the ones in blue. And again, I apologize that for some reason on my image, I only included one strand of DNA. It's a rookie mistake. They both should have been here. And then in our two daughter DNA molecules, aside from the original parent strand of nucleotides, we also have the daughter strand of nucleotides. All right, y'all, that wraps up this video. In the next video, we are going to focus on the itty bitty gritty nitty, I'm running out of itty words, the tiny details of DNA replication. We're going to introduce what I call the key players, the enzymes responsible for actually building this new strand of DNA that we've been talking about. Correction, this new strand of nucleotides that will make up the DNA molecule. All right, get ready. It gets exciting. All right, y'all, let me know if you have any questions.